Okay, so last time we talked about the phospholipids as the primary component of membranes and how they create that bilayer. Today what I want to do is or in this lecture what I want to do is run through the role of proteins within the context of the membrane. And membranes come in a variety of, or have a variety of proteins in them. As far as a structural level goes, the first kind of membrane that we have are what are called integral proteins. These are guys that are actually integrated and embedded into the phospholipid bilayer. So most of these guys are going to go all the way across, and so that you're going to have an outer portion, an inner portion relative to the cell. So one side is going to actually interact with the cytosol, one side is going to interact with the extracellular environment, and those proteins are what are going to be known as transmembrane because they go all the way across the membrane. And there are some integral proteins that are just going to embed it in one leaflet, meaning that they would work on one side or the other. And so they're embedded so they can't get away, but they do not work to connect the outside with the inside. Their function would be, I guess, fixed to one side or the other. So here's a figure you've probably seen before. You got the phospholipid bilayer that we talked about last time. And so now we've got these integral proteins, some that are transmembrane that go all the way across, some that are just embedded in one layer. And of course, the direction's important. So these guys are there's going to be one side that interacts with the outside of the cell. There's going to be one side that interacts with the inside of the cell, the cytosol. And the other kinds of things that one finds in here are going to be usually attached to. So notice we're going to have some other components, and we'll also have what are called conjugated molecules, where it's actually two different kinds of molecules that are attached to each other that also are going to be found inside this membrane. So one pretty abundant kind of conjugate molecule are what are called lipoproteins. These would be proteins that they're not embedded, but they are attached to the membrane in such a way that there's a lipid portion that is embedded, and then the protein is actually on the surface. And you would also have regions of an integral protein that are hydrophobic, and those would of course contain mostly hydrophobic amino acids. We talked about the nature of amino acids and how they would be found where you would need them to function. Regions that face into the aqueous environments certainly going to have lots of hydrophilic. So that would be polar, that would be the acidic and basic with the charges. Those guys would be very comfortable in that aqueous environment. So here's a figure of a protein that goes all the way across, and all of these amino acids that are in red are actually hydrophobic amino acids, so you can see the high level or number of hydrophobic amino acids that would be found in that area. And then here you would find a lot of charges that are interacting with the aqueous environment and those hydrophilic heads of the phospholipid. And of course the protein would have much more folds and so on. You can kind of see it there on either surface. But this just shows you the connection between the property of the amino acid and the fact that where it's located needs to match. The other proteins that are associated are what we call peripheral proteins. These are generally going to be on the inside because they would come loose if they're outside. These are not going to have a hydrophobic zone because they're not actually embedded in the membrane, and they're actually not part of the membrane itself. They are associated with the membrane, and that is where they hang out and do their work, but they tend to interact with the phospholipid surface, or they may actually be interacting and helping integral proteins, because obviously an integral protein can't come out of the membrane and go somewhere else in the cell to do its job. So these peripheral proteins are associated with these integral proteins, and in some cases will interact on the surface. So you can definitely see here these peripheral proteins as they interact with the inside of the integral proteins. And here you can see a couple of other peripheral proteins interacting here. and sometimes with proteins or phospholipids. The other component that we find in quite a little bit of abundance is these carbohydrates that are attached usually on the outside of a cell, and these tend to be attached either to lipids or proteins because they certainly would not be comfortable inside the cell. They would have way too much polarity to them and way too soluble in water. And these carbohydrates will be attached to a lipid component to form a glycolipid, or they may be bound to a protein to form a glycoprotein. And if we go back to that initial picture, that would be these guys. So you can see a glycolipid there, glycoprotein here, and it's usually going to be a dozen or so sugars, certainly more. So we got monosaccharides, disaccharides, and then anything from about 3 to 20 would be what we call an oligosaccharide. So these little chains of sugars, and a lot of them will branch and take on some fairly complicated structure, are going to be found on the surface. And as we'll see later, these guys they're almost like labels that are on the surface of the blood. And, I mean, sort of the cells. I was thinking about blood cells because if you think about the AB blood groups that humans have, those blood groups are actually associated with sugars on the surface. So A blood cells are going to have a certain kind of sugar, B blood cells have a certain kind of sugar, and O blood doesn't have any sugar, and AB blood have both sugars. So those sugars would be attached to the surface of red blood cells, but every cell is going to have different kinds of sugars attached to them and showing on the outside. Notice these are on the outside of the protein, and so these are going to be very much involved in how cells recognize each other. They're going to tell who their neighbors are, who am I related to, so who do I need to, if I have the same sugar as the one next door, do I need to do a certain thing? If I don't have the same sugar, do I need to do a different thing? So these are really going to play a role in how I recognize the cells around me, and as I recognize those cells, it's going to determine who I become and function inside the embryo. All right, so next time we will basically tie all this together and summarize some of the functions, and then we'll be done with the plasma membrane.